I'm Malix, and this is my meiosis presentation. To talk about meiosis, we must first talk about interphase. Interphase is the time in the cell cycle leading up to the M phase. The M phase in this presentation means meiosis, but it could also mean mitosis because interphase is the same for meiosis or mitosis, the time before it undergoes those processes. Meiosis is a process undergone by reproductive cells, while mitosis is a process only ever undergone by somatic body cells. So for this presentation, the process of M just mean meiosis. Interphase comes to several parts, including G1, the first growth phase, S, the synthesis phase, and G2, the second growth phase. Interphase is important, and its purpose is to grow and perform cell functions, to duplicate DNA to prepare for M, meiosis, and it takes the 96 chromatids to 92 chromatids. This happens in the S phase, the synthesis phase, when the DNA is duplicated. Meiosis starts with prophase 1, then metaphase 1, leading into anaphase 1, until telophase 1, followed by the process of cytokinesis. These processes together are referred to as meiosis 1. The distinction of 1 is important. Meiosis 1, prophase 1, metaphase 1. The 1 distinguishes between the two main parts of meiosis, the two main splits. So when talking about meiosis, it's important to refer to them as 1 and 2. Meiosis 1's separates homologous pairs. It creates genetic diversity with crossing over, and it takes the 92 chromatids to 46 chromatids per cell. Starting with prophase 1, what happens in prophase 1 is the nucleus dissolves, the chromosomes condense, and the process of crossing over occurs. The nucleus has to dissolve to free the DNA and the chromosomes to move. The crossing over is important for genetic diversity, and how it happens is the homologous pairs line up, and the homologous pairs are the identical sister chromatids matching up with other identical sister chromatids, and they match up with the sister chromatid pair that match in size and content, meaning they have the same genes. Now, when two parts with the same genes meet, they can sometimes break off and fuse with the opposite pair. This happens because, for all intents and purposes, they're identical. They have the same genes on them, so they can switch, and this creates genetic diversity. Next is metaphase 1. In metaphase 1, the homologous pairs line up in the center of the cell, and the spindle fibers attach to the centromeres. Next is anaphase 1. In anaphase 1, the homologous pairs are taken apart and drawn to separate ends of the cell, towards the centrioles, which is where the spindle fibers come from. Next is telophase 1. Telophase 1 is where the cell starts to split a little bit. The nucleus starts to come back a little bit, and you can start to see two distinct cells starting to form. In cytokinesis, the cells fully split, the nucleus starts to come back a bit more. Next, we go into meiosis 2, starting with prophase 2, leading into metaphase 2, leading into anaphase 2, until finally telophase 2, followed by the process of cytokinesis. All in meiosis 2. This is the second part of meiosis. This is the second main split of meiosis. We first go from 1 to 2 cells in meiosis 1. In meiosis 2, we go from 2 to 4 cells. In meiosis 2, we go from 46 chromatids to 23. We go from diploid cells to haploid cells. meaning they're missing their pair. But overall, Meiosis 2 is pretty similar to Meiosis 1. In Prophase 1, it's pretty much the same as Prophase 2. It's pretty much the same as Prophase 1, but without the crossing over, because that's already happened and they're not with their homologous pair anymore. Next up is Metaphase 2, in which the sister chromatids line up in the center of the cell and the spindle fibers attach to the chromatids in order to be torn apart in Anaphase 2. In Anaphase 2, the sister chromatids are separated by the spindle fibers and pulled to opposite sides of the cell. Much of this should sound similar to how it occurred last time in Meiosis 1, because it's pretty much the same. It's very, very similar. So you can pretty much guess what happens next in Telophase 2, when the cells start to split again. The nuclei starts to reform a bit, and it's important to note that there should be 23 chromosomes in each nucleus, and each of these starting to look a little bit distinct cells, not quite, but there should be 23 chromosomes. In cytokinesis, these cells fully split. And again, there are 23 chromosomes in each. These fully split cells with 23 chromosomes are gametes. They are reproductive cells. They are egg and sperm cells created at the end of meiosis. And they are created in the testes and ovaries, depending on if they are male or female. Reproduction happens when an offspring can start to form when the sperm cell and its 23 chromosome meets and fertilizes the egg cell with its 23 chromosomes. What does this actually look like? What does any of these processes mean? And we can maybe look a little bit deeper into that by tracking one chromosome through this process. Starting with interphase. Interphase G1. The cell gets bigger and more complex. It doesn't just grow, it also develops. The amount of organelles and diversity of them increase with the size. So in this image, it's difficult to see the chromosomes, but we're gonna pretend that we can see them very distinctly. So the star can mark about what we're trying to track. And it goes from here to here. Keep an eye on the star, as that's going to be the chromosome we're tracking all of this happening to. Moving on to the synthesis phase. The DNA duplicates itself, and we start to have two sets in the cell. Now, we actually don't go from 46 chromosomes to 92 chromosomes. We go from 46 chromosomes to 46 chromosomes. How does this happen? How does that work? If the DNA was duplicated, how do we have the same amount of chromosomes? Well, the trick is we have the same amount of chromosomes, but a double the amount of chromatids, because we count chromosomes by the centromere. And so, while one has double the chromatids, they have the same amount of centromeres. So mind the star, where it shows the chromosome we are actually tracking. The specific chromatid is the part of it to get as specific as possible. In the second growth phase, we see the cell gets bigger still. Too big, so it needs to split. Meiosis 1, the first part of meiosis. Prophase 1. We'll track one of the sister chromatids in this homologous pair. So this homologous pair should have 4 chromatids. And the star isn't very distinct in this image, but you know we're tracking 1. So in this, part of this chromatid crosses over with its homologous pair, And you could see that happening a little bit at the bottom. In metaphase 1, it lines up in the center. In anaphase 1, it is torn away from its homologous pair, and it's moving to an opposite side of the cell. In telophase 1, it's starting to have the nucleus reform around it a bit, and it's starting to be separated fully now, be fully isolated from its homologous pair in a completely different cell. In cytokinesis, this actually happens. The cell is fully split, and it is alone without its homologous pair, but it still has plenty of sister chromatid pairs around it, though none its actual match. And it still has its own sister chromatid, as a note. Next up, we have meiosis 2, the next split. You can see the sister chromatid and the chromatid we're tracking. And it starts up as before. The nucleus starts to dissolve a bit, and we move on to metaphase 2, where it lines up in the center, where it's about to be torn away from its sister chromatid in anaphase 2, where it is torn away, and it's taken to an opposite side. In telophase 2, it starts to split a bit from the cell containing its sister chromatid. And in cytokinesis, it is fully split away now. We can see that it is alone in this. This is not true. There is 23 other chromosomes in the cell with it, this gamete. Thank you for listening to my presentation. This is the end.